Have you ever thought about what's actually in your blood? It's like a bustling city just teeming with millions of different molecules all rushing around. Now, imagine you had to find one specific person in that entire city. How would you even start? Well, that's a huge challenge scientists face every single day. And today, we're going to see exactly how they crack that code hidden deep inside our blood.
And we're going to dive into this whole topic by focusing on one single, really high-stakes question. A doctor has a patient, and they need to know right now, is the medication they've prescribed actually helping, or could it be causing harm? The only way to find out is in their blood, and honestly, the clock is ticking.
So let's meet our patient. He's a 65-year-old man, and he's on a really powerful heart medication called amiodarone. Now, here's the tricky part about this drug. The dose has to be perfect. Just a little too little, and it's not going to work. But a little too much, and it can actually be toxic. The line is just incredibly thin. The only way for doctors to know for sure what's going on is to analyze a sample of his blood. And that brings us from the hospital right into the science lab. We've got our patient's blood sample, that's our haystack, and now the mission is to find that one single tiny little needle, the amiodarone molecule, hiding amongst millions and millions of other molecules. So this is the fundamental question, right? How in the world do you pull out just one ingredient from a complex chemical soup like blood? I mean, you've got proteins, salts, sugars, you name it. Well, the answer lies in this absolutely brilliant technique of separation. And the tool for this job? It's called chromatography. The name literally means color writing. Kind of a cool backstory, because the very first experiments used it to separate colorful pigments from plants. But at its heart, chromatography is the art of untangling a mixture. It basically works by making all the molecules run a race, knowing that different ones are going to run at totally different speeds. Okay, so this molecular race has two really key parts. First, you've got this stationary phase. And yep, it's exactly what it sounds like. It stays put. Then you have the mobile phase, which is a fluid that moves. It flows over or through the stationary part, and it carries our sample along for the ride. You can think of it like a bunch of runners on a crazy obstacle course. The mobile phase is the wind at their backs, pushing them forward, but the stationary phase is the terrain itself. Some parts are smooth pavement, but other parts are like super sticky mud. And this is how it all comes together. First, we inject our blood sample into that mobile phase. Then, as it starts to flow, the real magic, the separation, begins. Molecules that are more attracted to the stationary phase, our sticky mud, they get held back. They move really slowly. But the molecules that prefer the mobile phase, they zip right through, like they're on clean pavement. Over time, all the different components spread out, and at the finish line, a detector clocks each one as it crosses. It's brilliant. Now, for something as important as a medical diagnosis, we can't just use any old version of chromatography. No, we need the supercharged, high-tech version of this whole process. We need to bring in the big guns. And that's where high-performance liquid chromatography, or just HPLC, comes into play. The key words here are high performance. And that performance comes from using a really powerful pump to blast the mobile phase through the stationary phase at incredibly high pressures. We're talking up to 400 times normal atmospheric pressure. This makes the whole separation way faster, way more efficient, and unbelievably precise. So why is HPLC the perfect tool for our patient situation? Well, a few huge reasons. For one, it's versatile enough to handle delicate biological molecules like amiodarone without destroying them. It's also so sensitive it can find the absolute tiniest traces of the drug. But most importantly, it gives you incredibly precise and reproducible results, which is totally non-negotiable when a person's health is on the line. It's the gold standard, period. Okay, so the HPLC machine has worked its magic. It's separated out all the different components in the patient's blood, but here's the thing. That separation is totally invisible to the naked eye. The machine just spits out data, and we need to know how to read that data to get our diagnosis. The result that we get is a graph, and it's called a chromatogram. It basically looks like a skyline, a series of peaks on a timeline. And each of those individual peaks, that represents one specific substance that got separated out from the original mixture. So how do you make sense of this graph? Well, it all boils down to this one simple, powerful idea. The position of the peak on the timeline, when it shows up, tells you what the substance is. That's its unique chemical fingerprint. And the size of the peak, specifically its area, tells you how much of that substance you've got. Simple as that. But wait, how do we translate that peak area into a real-world number, like a concentration in the blood? Ah, for that you need a sort of secret decoder ring. It's called a standard curve. See, before running our patient sample, scientists run a bunch of other samples with known pre-measured amounts of the drug. They plot those results, and that creates a reference graph that gives them a direct link between the area of a peak and its actual concentration. All right, we have everything we need. We got the science, we have the tools, we know how to read the data. It is time to go back to our patient and solve this medical mystery once and for all. Okay, so here's what we've got on the table. The HPLC analysis gave us a peak for amiodarone with an area of 3,250 units. We also have that all-important equation from our standard curve. And finally, we know from the medical guidelines that the safe and effective range for this drug is somewhere between 0.5 and 2.5 micrograms per milliliter. Now comes the fun part. We just plug our patient's number into the equation. So 3,250 equals 1,500 times the concentration plus 250. Now we just do a little bit of simple algebra. We subtract 250 from both sides, that gives us 3,000, then we divide that by 1,500, and we get our unknown concentration. And there it is. The calculation shows that the concentration of amiodarone in our patient's blood is exactly 2.0 micrograms per milliliter. So, what's the final verdict? We take our patient's result, 2.0, and we compare it to the safe range of 0.5 to 2.5. And what do you know? It falls squarely inside that therapeutic window. The answer to our big question is clear. The patient's medication is at a level that is working effectively, but it is not yet toxic. The doctor can breathe a sigh of relief and continue the treatment with confidence. So we solved the case for this one patient, which is fantastic. But the power of this technology, it goes so far beyond just a single hospital room. This science of separation is like a silent hero working behind the scenes in so many areas of our lives. I mean, just think about it for a second. Chromatography is used to make sure the medicines you take are pure and have the right dosage. It's used to check for dangerous pesticides or toxins in our food. It helps scientists measure pollutants in our water and soil. And in forensics, it can analyze tiny, tiny traces of evidence from a crime scene to help catch a criminal. It's truly a fundamental tool for understanding the world on a molecular level. We just saw how this technique can find one single molecule in a tiny drop of blood to save a life. It really makes you wonder, doesn't it? What other secrets are hiding out there in plain sight? In the food we eat, the water we drink, the very air we breathe. Just waiting for the right tool to come along and bring them to light. Thanks so much for watching.